Hi guys, this is Dr. Rajeshwar from yrpharmatube. In the preceding video, we learnt the citric acid cycle pathway. If you did not watch the video, hit the I button on top right of this video to watch it. To watch the other topics of carbohydrate metabolism and other subjects of pharmaceutical sciences, click on the links given right below this video. In this lesson, we shall try to understand the energetics, significance, regulation and fate of citric acid cycle. The citric acid cycle is a central pathway for oxidation of carbohydrates, lipids, and proteins. The pyruvate produced from the glycolytic pathway enters the citric acid cycle, which undergoes oxidation to acetyl-CoA by the pyruvate dehydrogenase complex. This three enzyme system uses five components NAD+, coenzyme A, FAD, lipoamide, and thiamine pyrophosphate. Each turn of the citric acid cycle involves entry of two carbons as the acetyl group of acetyl-CoA and loss of two carbon atoms as carbon dioxide. Acetyl-CoA condenses with oxaloacetate to form citrate. After one turn of the cycle, oxaloacetate is regenerated to begin the process anew or another cycle. As a result of oxidation of acetyl-CoA in the citric acid cycle, three molecules of NADH, one FADH2 and one GTP are produced. The table reviews the energetics that have been accomplished in one turn of the citric acid cycle. The reducing equivalents NADH and FADH2 are oxidized by respiratory oxygen. Oxidation of each NADH in respiratory chain generates 3 ATP molecules. Thus, 3 molecules of NADH generate 9 ATPs, while oxidation of 1 FADH2 in the respiratory chain generates 2 ATP molecules. One molecule of ATP is formed at the substrate level during the conversion of the succinyl coenzyme A to succinate. Thus, a total number of 12 ATPs are generated from one molecule of acetyl-CoA. Under aerobic conditions, complete oxidation of glucose occurs in three stages. They are glycolysis, pyruvate dehydrogenase complex, and citric acid cycle. Total yield of ATP during oxidation of glucose is obtained by adding ATP generated at each stage. ATP generation during oxidation of glucose is given in the table here, as glycolysis produces 8 ATPs, pyruvate dehydrogenase forms 6 ATPs, and from the citric acid cycle 24 ATPs, because glucose gives rise to the formation of two molecules of acetyl-CoA. Aerobic oxidation of glucose yields 12 into 2 is equal to 24 ATPs at this level. Thus, aerobic oxidation of glucose generates a total of 38 ATP molecules. However, the amount depends on the shuttle used in the transfer of the reducing equivalents from cytosol to mitochondria. Role of vitamins in citric acid cycle. 5 B vitamins such as riboflavin, niacin, thiamine, lipoic acid and pantothenic acid are associated with the citric acid cycle, essential for yielding energy. Riboflavin in the form of FAD, that is flavin adenine dinucleotide, a cofactor for succinate dehydrogenase enzyme. Niacin can be used as a thiamine diphosphate, which is required as coenzyme for decarboxylation in the alpha-ketoglutarate dehydrogenase reaction. Thiamine as a thiamine diphosphate is required as coenzyme for decarboxylation in the alpha-ketoglutarate dehydrogenation reaction. Lipoic acid is required as coenzyme for alpha-ketoglutarate dehydrogenation reaction. Pantothenic acid is employed as a part of coenzyme A, the cofactor which is attached to active carboxylic acid residues such as acetyl-CoA and Succinyl-CoA. Significance of citric acid cycle. The primary function of the citric acid cycle is to provide energy in the form of ATP. In other words, the citric acid cycle produces reduced coenzymes that enter the electron transport chain and ultimately produce ATP. Citric acid cycle is the final common metabolic pathway for the oxidation of carbohydrates, lipids, and proteins as glucose, fatty acids, and many amino acids are all metabolized to acetyl-CoA or intermediates of the cycle. Citric acid cycle has a dual function. It functions in both catabolism and anabolism. In the liver, intermediates of TCA cycle are used for the synthesis of fatty acids, cholesterol, amino acids, porphyrins, and glucose. In some liver diseases like hepatitis and cirrhosis, amphibolic role of citric acid cycle is affected. Some metabolic pathways end in the constituent of the citric acid cycle, while other pathways originate from this cycle, such as gluconeogenesis, transamination, fatty acid synthesis, and heme synthesis. Gluconeogenesis. All major members of the citric acid cycle, from citrate to oxaloacetate, are glucogenic. They can give rise to glucose by gluconeogenesis. Transamination. Oxaloacetate and alpha-ketoglutarate serve as precursors for the synthesis of aspartate and glutamate, respectively, by transamination reaction, which in turn are used in the synthesis of other non-essential amino acids, purines, and pyrimidines. Fatty acid synthesis. Mitochondrial citrate is transported to the cytosol where it is cleaved to provide acetyl-CoA for the biosynthesis of fatty acids and sterols. And lastly, heme synthesis. Succinyl-CoA, which is the intermediate of TCA cycle, together with glycine, is used for the synthesis of heme. Now, coming to the regulation of citric acid cycle. The citric acid cycle is regulated at 4 different steps as shown in the figure, which are irreversible reactions. Number 1, conversion of pyruvate to acetyl-CoA. Number 2, synthesis of citrate from oxaloacetate and acetyl-CoA. Number 3, oxidation and decarboxylation of citrate to alpha-ketoglutarate. And number 4, conversion of alpha-ketoglutarate to succinyl-CoA. Conversion of pyruvate to acetyl-CoA. The pyruvate dehydrogenase complex is inhibited by high concentrations of ATP, acetyl-CoA, and NADH. The presence of these compounds in abundance signals that the cell has an adequate supply of energy and thus energy metabolism is slowed. Synthesis of citrate from oxaloacetate and acetyl-CoA. The enzyme citrate synthase is an allosteric enzyme that catalyzes the conversion of oxaloacetate to citrate, that is in the step 2 of the citric acid cycle. Since it is an irreversible reaction, it is a suitable control point for regulation of the entire cycle. ATP inhibits the enzyme in vitro, but significant changes in the ATP concentration are unlikely in vivo. Therefore, ATP may not be a physiological regulator. Some bacterial citrate synthases are activated by alpha-ketoglutarate and inhibited by NADH. Oxidation and decarboxylation of isocitrate to alpha-ketoglutarate. Mammalian isocitrate dehydrogenase enzyme is allosterically activated by calcium ions and ADP and inhibited by NADH. In mammals, the enzyme is not subject to covalent modification. In bacteria, isocitrate dehydrogenase enzyme is regulated by phosphorylation reaction. ADP is a signal that the levels of ATP must be low and therefore the rate of the citric acid cycle should be increased. Interestingly, isocitrate dehydrogenase is also inhibited by high levels of NADH and ATP. Conversion of alpha-ketoglutarate to succinyl-CoA. The alpha-ketoglutarate dehydrogenase complex resembles the pyruvate dehydrogenase complex. The enzymes have quite different regulatory features. No kinase or phosphate is associated with the alpha-ketoglutarate dehydrogenase complex. Instead, the alpha-ketoglutarate dehydrogenase complex is inhibited by high levels of the products of the reactions that it catalyzes, namely NADH and succinyl-CoA. NADH and succinyl-CoA are inhibitors of the alpha-ketoglutarate complex in vitro, but it has not been established that they have a significant regulatory role in living cells. It is further inhibited by high concentrations of ATP. Lastly, let us look at the fate of carbon in the citric acid cycle. Acetyl-CoA entering the citric acid cycle is highlighted in blue color in the figure to show the fate of its two carbon atoms through reaction 4. After reaction 5, the carbon atoms contributed by the acetyl-CoA acetyl group from this turn of the cycle are no longer highlighted because succinate and fumarate are symmetrical molecules. Thus, C1 and C2 become indistinguishable from C3 and C4 beyond this point in the cycle. Carboxyl groups that leave the cycle as carbon dioxide in reaction 3 and 4 are shown in green color. Note that these departing carbon dioxide groups derive from the two oxaloacetate carboxyl groups that were incorporated as acetyl-CoA in earlier turns of the citric acid cycle. This is the list of references followed for the lesson. That's all in this video, the energetics, significance, regulation and fate of citric acid cycle. In the next video, we will discuss about the HMP shunt. Till then, never stop learning and never stop watching my videos. Thank you for watching this video.